welcome to our other expert, Dr. Jenny Tamayo. She is Senior Science Research Specialist at Department of Science and Technology, DOST, FPRDI, Philippines. She'll discuss about the utilization of forest-based materials during this pandemic crisis. So we welcome you, Dr. Tamayo. She will deliver her talk on this one, on utilization of forest-based product. I also want to welcome to Dr. Ashok. He is Senior Research Scientist at Kansas University Medical Center, Kansas, USA. He will discuss about male sexual dysfunction and insight into screening. We welcome you. I extend my welcome to all participants from India, Philippines, and USA. So I welcome you all on the behalf of Department of Pharmacy. I hope this event will be a grand success. So thank you. Thank you all.
Thank you, ma'am. Thank you so much. The aim of education is the knowledge not for found but for values. It is an occasion like this we get opportunities to test our knowledge and understanding. We look forward to get an exposure about what the best of the brain think about these very issues. And for the same, I would like to invite Dr. Hitesh Kumar Agarwal, presently working Chair-elect ACS Coastal Georgia and also as Assistant Professor Pharmaceutical Sciences, School of Pharmacy, South University. He was professional and teaching experience of more than 15 years. He has completed his Ph.D. in medical chemistry from University of Rhode Island, Kingston, RI, under guidance of Dr. Canvas Frank. His Masters of Pharmaceuticals Chemistry from Hamdard University, Delhi, India, and his Bachelor of Pharmacy from Meerut University, India. He is awarded with many awards like SEPRA, South University Excellence in Pharmaceutical Research Award in year 2017, co-investigation under project title Targeted Doxorubicin Delivery to Glioblastoma by CC Leader Development Award for January 2012, ACS Leadership Institute, Texas, William Porter Prize 2005-2006, University of Rhode Island, Graduate Research Assistantship 2004 to 2005, USAID, CONDRAD, AICTE Scholarship 2002 to 2004, All India Council of Technical Education. He has one US patent. He has credited to more than 50 national and international articles. So I would like to invite you for few words.
Thank you very much, Shweta. That was a very nice introduction. Thank you very much, Dr. Verma, for organizing this event. And it is a very pleasure for me to share this stage with Dr. Tamayo and Dr. Bhalotia. In fact, my research career gets a very good credit to Dr. Bhalotia because I am in this country because of him. My PhD started because of him. And it just happens to be the fate that now we are sharing a stage. And it is so nice to see you one more time. So when you were sharing all those information about me, it felt like you were talking about someone else. Did I do all that? But believe me, that's one step at a time. Take a small step and keep taking another small step after that. And it will guide you, right? So today I think we are going to be talking about sexually transmitted diseases and infection. And it just happens to be the topic of my PhD. So when Dr. Verma approached me, I thought like this is the best place for me to share my work. So I'm going to be sharing my presentation. Is it a good time now, or are we okay?
Yes, sir, you can start.
All right. So I'm going to share my presentation here with you. Can you all hear me very well?
Yeah.
Okay. All right. So I came in to complete my PhD at University of Rhode Island College of Pharmacy, and before that I had done my M.Pharmacy in pharmaceutical chemistry from Hamdard University, as you mentioned. So when I came here, I had very limited knowledge of synthetic organic chemistry, and I was very scared in the beginning actually. So I just wanted to succeed in life and learn from the best. And it just happened to be in I went to the lab where they were working on very simple project, very simple project, but a great idea. And with this short period of 25 minutes of presentation, I will share what I have done. Um, it's difficult to complete it, but the main important aspect is that these are very simple structure molecules. These are just simple esters. And all these derivatives that I synthesized over a period of 4.5 years of my PhD, I got several publications out of it because idea was very strong. What was the idea? Idea was that this work started with my advisor. He synthesized some conjugates in 97, 96, 97. That was the time when HIV was one of the scariest topic. It was very well spread around the world. There was no control, and it was life threatening at that time. Drugs were not there to cure the disease. Now HIV can be cured, but it is still spreading at a very slow rate. It is still continuously spreading. And what is the best way to get rid of it? Prevention is the best cure. Prevention is even better than the cure, right? If you prevent that viral transmission, virus is not going anywhere. It will just fade away. It will just die on the surface, right? So this project included conjugating nucleosides with fatty acids. Now these fatty acids are special. They are not just any cooking oil. They are specific fatty acids. The goal was to improve the activity of nucleosides, which were main molecules, and their activity was enhanced by using some specific type of fatty acid. And for that purpose, we used initially several types of fatty acids, and the screening library reduced that number to 3, and various types of nucleosides, various types of permutation and combination. The goal of the work was to improve the lipophilic nature of these nucleosides. Nucleosides, I hope you remember that they make the part of DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So these are the nucleosides which make these molecules, and virus relies on proper synthesis of these molecules in the body. So if you prevent the synthesis of these molecules, it won't work. So our goal was to improve the presence of nucleosides in the cell. So if you improve the lipophilic nature of a molecule, it will penetrate the cell membrane better, so cellular uptake will improve. And there were two molecules, one was nucleoside, another was fatty acid, and they were targeting two different virus life cycles, two different stages of virus life cycles. And then we continued our work in different areas. We used to study whether it is affecting on the sperm, if it is active against the virus, because all these molecules were targeted for what? Preventing the viral transmission. So it was basically offering an extra layer of protection, which was already there during sexual intercourse. But if these molecules were used in that unprotected sex or any form of transmission where there is a possibility of virus to contaminate the other person, using these molecules will prevent that viral transmission. So it was imperative to study the effect of these molecules on virus as well as on sperm. And we also studied how much molecule was going inside the cell. So just giving you a brief overview, most of the virus, they target the membrane by binding with the membrane through certain specific type of proteins certain type of protein on the virus target the certain type of protein on cell membrane, and then the virus will fuse. This is same for COVID as well. The virus will go inside the cell, then the genetic material will enter itself. The viral genetic material will enter itself into the DNA. It will incorporate inside the nucleus. It will bind with DNA and RNA, and then the proteins will synthesize. Nucleic the genetic material will be replicated. Some of the viruses are RNA dependent, some of the viruses are DNA dependent, whichever it may be. All that is synthesized, and gradually they start to move towards the membrane. Well, protein is hydrophilic. It tends to stay inside the fluid of the cytoplasm. It doesn't like to go towards the membrane because membrane is hydrophobic, proteins are hydrophilic. So an important process happens in the virus maturity, that is, the protein which is synthesized in this process gets converted into lipophilic molecule by addition of fatty acid on those proteins. So they become hydrophobic, and then they start to move towards the outer cell membrane area, so that the genetic information and the proteins, once they are converted into hydrophobic molecules, they can move towards the membrane so that the virus particles can be synthesized. One virus goes in, hundreds and thousands of thousands of them comes out, right? So this was the path life cycle. Our molecules contain a nucleoside analog which was working on reverse transcriptase enzyme, crucial for genetic information to be transported to the nucleus. Another part was this fatty acid, which was binding or preventing this particular step. The post-translational modification of the proteins. Both of these steps were two different pathways, and both of them were targeted. In addition, the molecule became more hydrophobic so that it can enter inside the cell much easier. Hydrophobic molecule can cross the cell membrane better, right? So in simple terms, we started with attaching the fatty acids with a nucleoside. So nucleoside has an alcohol, and acid have carboxylic acids. You combine them, you get an ester. So this was a starting molecule, a simple ester of various types of nucleosides, FLT, D40, lamivudine, and then stavudine, and then FTC. All these ampicillin. So all these molecules were attached directly with the fatty acid, and these simple esters were synthesized, purified to the highest purity, tested against the virus. Some of them were very complicated to synthesize because there were two different type of functional groups. So a lot of direct synthesis or the protection chemistry or deprotection chemistry is involved. Now, some of you want to be synthetic organic chemists, you want to go for medicinal chemistry, you will have to learn how to do the synthesis. There will be a stage where you will have to do a lot of multi-step synthesis. Now, this is just 5-step synthesis, but it can even go up to 10-step, 15-step, things like that. So these were simple reactions, but a lot of protection, deprotection chemistry was involved before the fatty acids could be attached on either the alcohol or the amine. So on alcohol or the amine. But we got them, and the mechanism of action was that these conjugates, which being more lipophilic, will go inside the cell where, let's say, there is a virus particle here. Okay, let's say there is a virus particle. It entered through the sperm, along with the sperm. It entered. So the drug, if the drug is used at that time, along with the virus, the drug is used. So at that time, the drug will go inside the cells much faster because it is very, very hydrophobic. There, it will split into two halves, a small molecule of nucleoside and fatty acid. Okay? Now, Drug, which is nucleoside, will be converted into its activated form, and it will bind with the DNA or RNA, and it will prevent the transcription or translation depending on the targeted site. So the one of the important pathways prevented by inhibiting the reverse transcriptase enzyme. Now I'm giving you a broader picture. I'm not just talking about DNA here, because in HIV it is one type, but if you extrapolate that same concept to various types of viruses, it could be DNA or RNA. So that's why I'm saying that term DNA or RNA. But for virus HIV, it was only DNA. Okay. So reverse transcriptase was inhibited by nucleoside activated triphosphates. On the side, if these fatty acids were preventing this N-myristoyl transferase, this enzyme which attaches the fatty acids onto the proteins. So two different type of important enzymes were inhibited, which are important for two different parts of the life cycle of the virus. And all these molecules were targeted for topical application only. They were not designed for oral intake. They were only designed for use as the topical jellies. And they did enter into the preclinical studies. Of course, fate happened. They did not succeed in some of the areas, but they were very active. For example, if you look at the parent drug FLT, one of the nucleoside, we don't have to go into the structure, but just looking at the parent nucleoside, the drug itself, which was OH, it was not active against the cell-associated virus. It was active against the free virus, but once the virus goes inside the cell, it was not active. But if we conjugated it with fatty acid, it was not only active against the free virus, but it was also active against the virus which was inside the cell. So it was working. Here you can see that some of the parent nucleosides, such as 3TC, they are not active against, they are not very active. They are active, but they are not very active. In fact, they were developing drug resistance. Virus was developing drug resistance. That was a major challenge. It is even now, it is a major challenge. So when we conjugated them with fatty acids, they were active. In other case, it is very active against both free virus particles versus the one which is inside the cell. So if the virus goes inside the cell, it was very hard to deal with those type of viruses. They were working very fast, they were multiplying very fast, but these type of molecules were able to enter the cell and kill those type of viruses as well. So it was important to identify that they are working better. They are working better because they were working synergistically. One pathway was activating the other pathway. But how? Besides the synergistic effect, how were they working? So in order to test that, we attached them with the fluorescein, which is like a fluorescent molecule. So when a parent nucleoside such as FLT or let's say 3TC, these are the nucleoside names. There are several nucleoside names. When they were used, you can see that very small particles were able to go inside the cell. But when fatty acids were attached, the fatty acid esters of the nucleoside were attached, you can see the fluorescence is so high, means more and more drug is going inside the cell, whereas very small amount is going inside the cell. So this was the proof concept that it is working, and following that, we made various versions. What did we do? In previous cases, we only had one nucleoside attached with fatty acid. So we thought like, okay, let's attach another fatty acid at the end. Attach another nucleoside at the end. And we used various permutation and combination. Here you can see we use glutamic acid in basically amino acid. So this is one of the examples where fatty acids, multiple different type of fatty acids, such as you can see here, FLT, FLT, AZT, and 3TC. Three different type of nucleosides are attached with just one molecule. So with one molecule, you get three different type of nucleosides. What is the advantage? If one virus is resistant against 3TC, then FLT will work. If one virus is inactive against one type of nucleoside, then other type of analog will work against that virus. So drug resistance was easily handled, although this molecule was little bit difficult. It started with simple attachment of glutamate attached with nucleosides. I'm not sure how much you remember the structure of amino acids, but if you remember structure of glutamic acid, it contains two carboxylic acid and one amino group. So I attached one particular nucleoside on one carboxylic acid and another type of nucleoside on other carboxylic acid. So two different type of nucleosides were able to target, and on the amino group we attached third type. So one type of nucleoside, two type of nucleoside, and three type of nucleoside. Okay. What does this molecule contain? Three different type of nucleosides attached on one molecule. And when they compared with just one single nucleoside, such as if we were to see 3TC, it was noticed that the multi-drug resistant virus, here you can see MDR. So oops, why is this not working? I'm trying to use my pointer, yes. When only one particular type of nucleoside was tested, it was not active against multi-drug resistant virus. If you see FLT, that is also not active against multi-drug resistant virus. If you see AZT, that is also not active against multi-drug resistant virus. But when we combine all three of them in one molecule, that molecule, this molecule number 34, is active against multi-drug resistant virus. So when this molecule is used in jellies as a topical application, so that will prevent the virus more effectively. And as I said, prevention is better than cure, right? So a lot of work was done in 4.5 years, and through this work, I got more than 13 publications and one patent, including one in science. This, if you some of you may not remember, but this is one of the most reputed journal in research community, and there is still one publication left. I still have one more to go. It all started with one small step. I was sitting in RK Puram in Delhi on internet cafe, and Dr. Belotia would just happen to sit beside me, and he said, he noticed at me, he looked at my application, I was looking for universities, and he reached out to me, he showed me the path, he gave me the cover letter. I used that cover letter to apply for 45 universities. So that small step helped me get admission at University of Rhode Island. Thanks to Dr. Belotti, I got my admission. And I am always thankful to him for that particular step. So Mark Twain, he was a great American philosopher. He said that if you do the very first thing in the morning, eat a live frog. If you eat a frog, then you can go through the rest of the day with the satisfaction that you are already done with the worst possible thing that you could have done in your day. Everything else in the rest of the day is going to be very easy. I think that PhD was my eating my frog. You all can do that. You all have to do that. You all will need to do that at some point of your life. Eat that frog. What is your frog? Your frog is the biggest, most important task of your day or of your life. Focus on that instead of procrastinating. The longer you procrastinate, the longer you delay your success, which is supposed to be in your approach. And if you want to read this concept, read this book, Eat That Frog. It's a very small book, but very fascinating book. How to stop procrastinating and getting things done in less time. In simple words, you do your biggest and hardest task as soon as possible. And there is a success principle which every one of you can use in your life. Divide your works all the responsibilities that you have to do in week, month, or year into 4 quadrants. Convert that into quadrant 1 and quadrant 2, which contains the urgent and the most important work, and not urgent but important work. And focus on these two aspects: urgent but not important, not urgent and not important. These works should be avoided and never pursued. When successful people organize their life's activities in these 2 quadrants, they become successful. So I hope my small presentation help you in your journey. Um, if you have any questions, I'm more than happy to answer them.
Thank you so much, sir, for giving your valuable insight. This means a lot to us. Thank you so much. Now, I would like to invite.
If there is any query, any query, you can ask participants. Any query you want to ask Dr. Agarwal, you can ask. No? Okay. Thank you, Dr. Agarwal. Thank you so much.
Thank you.
Now, I would like to invite Dr. Jennifer P. Tamayo, present working as Senior Science Research Specialist, Department of Science and Technology, DOST, Forest Product Research and Development Institute, FPRDI, Philippines. She has completed her BS Chemistry from University of the Philippines, Los Banos, UPLS in year 1989 to 1993. MS Chemistry from UPLB in year 1999 to 2003. Masters in Management from Colegio de San Juan Letran, Calamba in year 1996 to 1998, and her Ph.D. Agriculture Chemistry minor on Chemical Engineering from University of UPLB in year 2007 to 2010. She has professional experience of more than 15 years. She has relevant work experience on forest product research and development shoot senior science research specialist from December 27, 2002 to till now. Forest product research and development shoot science research specialist from 1997 to 2002. Forest Product Research and Development Institute Research Assistant from 1994 to 1996. She is also membership in many professional organizations. Ma'am, I would like to invite you for a few words on employing science and the utilization of forest-based material amid the pandemic crisis.
Okay, good morning to everyone. It's so nice to be invited in your National Science Day today. So I'm going to present one of our research endeavors in our country. So this presentation is all about exploring science and utilization of forest-based materials amid the pandemic crisis. Okay, next slide, please. Okay, so I have searched about the National Science Day, and this is the theme for 2021. So I hope that I could spread a message about the importance of science used in the daily life of the people, and this is my goal for this presentation, and also the development and technologies in the field of science. So next slide, I'm going to present our institute. Okay, the UST FPRDI, Department of Science and Technology, Forest Products Research and Development Institute, has this mandate to conduct research and development on wood and non-wood forest products, transfer technologies, and provide technical services and trainings. So, I'm not gonna read the mission. Okay, so we have the COVID-19 research initiatives in our institute, and this is the worldwide scenario. This is as of February 1, 2021, and we have a 102 million confirmed cases and 2.2 million confirmed deaths. And we all know this is from WHO data. We all know that this became a pandemic last year, the COVID-19 pandemic. And for the next slide, we have our scenario. Yes. So in our country, we have the total cases of 574,247 already as of February 27, as of yesterday. So I have not captured the Indian scenario in this presentation. So the global outbreak was due to the novel infectious disease caused by severe acute respiratory syndrome coronavirus, and was declared a pandemic on March 11, 2020, last year, by WHO. So these are just the data of our reported cases as of February 27, 2021. Okay, so next will be the needs. I think the needs. The next slide, please. Okay, sorry for I have poor internet connection. So I have let Dr. Amita share the screen. Next slide, please. Okay. Can we have the next slide? Can you still hear me? Am I still online?
Yeah.
Okay, okay. Can you please change the slide to the next slide? It's COVID-19. I'm finished with this. Okay. Oh, maybe there's a delay.
Which one you need? This one?
The next slide. The next slide. I'm finished in this slide already. It's not moving in my screen.
Yeah?
Yeah, it's not moving in my screen. Yeah. Can you?
But here it is showing.
Yeah. My slide is COVID-19, a global outbreak. So maybe the next slide. It's not moving.
Slide number?
I think this is number. Wait.
So right now we are seeing slide number 6. Maybe
yeah,
we can move
number
to slide
7.
number 7.
Yeah. Slide number 7. Sorry.
I'll share again.
Okay.
There's some problem.
Yeah.
I don't know what is the problem.
I thought I was lost in the connection.
Is it okay now?
The next slide.
Okay.
Yeah, okay. So this is just to show you our government is always reporting to us how many figures are there for the cases, confirmed cases, how many deaths, how many recovered cases. And I just want to emphasize on the figure on the bottom, the mask. We all know that we need to wear mask. Hugas means proper hand washing, and iwas means physical distancing. So this is this Tagalog words. So that we could continue our economic activities. Okay, thank you. Next slide, please. Okay, for the needs, when we have developed our products in this time of pandemic, we have this growing consciousness about health and risk of contracting the virus, and we all know today that there are still mutations and strains of other COVID-19 virus, of the coronavirus. And our goal is to minimize health risk and exposure to coronavirus. So this is the proper hand washing. And we are involved in forest-based materials, non-wood and wood materials. Okay, next slide. Okay, we have this innovative technology, and our country have this Republic Act 11469, or we call Bayanihan, the Philippines one. And in this endeavor, we have developed products such as liquid and bar soaps and antimicrobial hands from forest-based materials. And this will help augment prevention and protection of people against the coronavirus. And to tell you, we have started our development of these products during the time that we are all in total lockdown last year, sometime in March. So it's very difficult because we have enhanced community quarantine in our country. Maybe it's a different term in your countries. But this quarantine period, we have reported to work to develop products that could help our nation. So, okay, so as part of our response, so there's a continuous product development. Okay, next slide, please. So we have these completed researches from forest products. We have bamboo, and we have the bamboo kiln, and the bamboo charcoal. The bamboo charcoal is mainly used for fuel. However, because of this pandemic crisis, we have made this as main ingredient for our developed products. Also, next slide, please. We also have these forest-based materials like, as I have shown earlier, the bamboo charcoal, the bamboo activated carbon, and also the bamboo leaves are all waste materials. And we have this Manila enemy resin. The resin is the source of essential oil for the development of our products. These are all forest-based. Okay, thank you. Next slide, please. Okay, and these are the FPRDI developed products. We have the bamboo charcoal bar soaps and the bamboo activated carbon liquid soaps, and we have the bamboo hand mist. So we have developed these products for, I think, 2 months, and then this was reported by our DOST secretary, and we are continuously testing the materials for FDA approval and for the transfer of technology to our prospective cooperators. Okay, next slide. The next slide would be the bioefficacy testing. So I have my colleague here, microbiologist. She's here in the call, Ms. Rowena Ramos. So this is the well diffusion assay using the 4 indicator microbial strains. Of course, we cannot do the coronavirus strain. We have used the gram positive, gram negative, and fungi, mainly as responsible for the skin. So the clearing zone for bamboo charcoal, activated carbon, and I think this is Manila enemy. I can see from my screen. So these are the results of our bioefficacy testing, and there is a clear zone on the side of on the site of the inoculation of the samples of our samples. Okay, next slide, please. Okay, this is the average zone of inhibition, the minimum zone of inhibition in millimeters. And these are the antimicrobial products, and these are the test organisms that we have tried. So we have this legend, greater than 16 with antimicrobial activity, and we have intermediate. So all in all, all the products are shown in Bacillus subtilis, greater than 16. Also in E. coli for the bamboo charcoal liquid soap. So these products are also filed for IPR, for patent review. So are processing for this IPR issue. Next slide, please. Okay, I'm presenting the business opportunities for these forest-based antimicrobial products because we are in the new normal. There are many businesses that were shut down during the time of pandemic, and we see that this development of these products will help the livelihood of some of the people that were displaced because of this pandemic crisis. So the products for the new normal will be the personal care hygiene products, and is the proper hygiene to prevent the spread of disease, while using raw materials like bamboo that are sustainable, eco-friendly, and natural. And proper hand washing with soap and water will prevent the transmission. This is one of the minimum public health protocol for all the uh, prevention of transmission. Okay, next slide, please. Okay.
Yes, it's live.
Okay, so wait for the next slide. Uh-huh. Oh, it's still in this slide. As shown
Okay.
here, the proper hand washing. So this is the developed soap already in this picture. Okay, we're waiting for the next slide.
Okay, I'm just
Okay. Sorry, because
okay.
if I'm going to share this, maybe that's more of a
Okay.
problem.
Okay.
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 Uh,
Okay.
we'll
Wait,
wait.
wait, wait.
And this is slide number, I think this is slide 16. So slide 17. Okay, we will be in slide 17.
Okay.
Okay, yeah, there. Okay, so as you can see, this is just the principle of why hand washing with soap and water is very important and very effective. So imagine that this is your skin and the dirt is the brown ones, and the function of soap molecules as a micelle has polar and non-polar side, non-polar tails, and the polar head. So viruses such as coronavirus, influenza causing virus, Ebola, Zika have their genetic material encased in a layer of fat called the lipid envelope. And soap molecules are pin-shaped with okay, I cannot read. With hydrophilic and hypophilic portion. And being oleophilic, the tail portion of the molecule tends to have an affinity for F4 and competes with the lipids in the virus envelope. Okay, I think there's something missing there. And the tail competes with the band that binds the RNA in the lipid envelope, thus dissolving the virus into its components. And the water will take away all the dirt and in your skin. So that's the function of soap and water. So that's why it's a very effective, and it's included in the minimum health protocol. Okay, next slide. Okay, this is just to show you the safety data sheet, because in the preparation of the bamboo hand soap, we have the main ingredients, the bamboo charcoal or the activated carbon powder, and then coconut oil, palm oil, caustic soda, and the essential oils. So among these, caustic soda, this is the safety data sheet, because if we're going to teach this to our countrymen or fellow people in the countryside. So we are going to conduct training. We are responsible to inform them the safety of the chemicals that they are going to use. So this is the uh, safety data sheet for the caustic soda. Okay, I cannot read. It's very little. Okay, next slide. Okay. For the bamboo hand soap making, first we have to do the charcoal preparation. Actually, it's just like serendipity, because we have carbonized and we are making charcoal for fuel, but we have used this during the pandemic because this is the only available material that we have during that time. So we have converted these materials so that we could help prevent and augment in our own little way. So this is the typical soap making process. So it's illustrated here. Okay, next slide, please. Okay, so after the soap and water, now the alcohol-based hand sanitizers, how do they help get rid of coronavirus? So this explanations are shown here. When they have issued this minimum public health protocol for for the virus, uh, this is the scientific explanation, because all the people they just say, okay, you just wash proper hygiene, hand hygiene, just wash with soap and water, you use sanitizer in the absence of soap and water. So this is the main explanation for that. So like soap, the alcohol present in hand sanitizer dissolve the lipid envelope, thus inactivating the virus. So however, the alcohol does not come in contact with all parts of your hands, unlike soap and water. And the alcohol tends to change the shape or denature the mushroom shape protein structures that stick out of the lipid envelope. So I'm just explaining this in layman's term. It's not very deeper scientific explanation, but this is a representation of how this hand sanitizer and hand hygiene should be, why they help get rid of coronavirus. And to be effective, the sanitizer should contain at least 60% to 80% of alcohol. Okay, next slide, please. Okay, so these are the list of alcohol and non-alcohol compounds and commonly used for sanitizers. We have the alcohol-based, and we also have the alcohol-free. So the alcohol-based, 60 to 95% alcohol, either ethanol, isopropanol, or N-propanol, and we also have the hydrogen peroxide. So these are WHO, as per WHO guidelines. And alcohol-free, this antiseptic, the chlorinated ones, chlorhexidine, chloroxylenol, iodine, iodophores, quaternary ammonium compounds, and triclosan. So we all know that triclosan is not commonly used now. So in between these two groups of sanitizers, we have what we have called excipients. So we have glycerin, which will act as a humectant and fragrance, and sometimes colorant. So okay, next slide, please. Okay. For the hand mist making, this is for the bamboo. We have utilized the bamboo leaves, which are considered waste after harvesting the bamboo poles. So these are the raw materials. We have the food grade or cosmetic grade ethanol, 95%. Bamboo leaves, sodium benzoate for preservative, glycerol, hydrogen peroxide, and what else? Written there, I cannot see in this slide. Okay, so next slide, please. And for the safety data sheet, yes, because we have that intention of conducting training for this. We put the glycerol and the hydrogen peroxide. So hydrogen peroxide is an oxidizer, just to inform our community-based livelihood projects if they're going to use this technology for the new normal. They are well prepared for what are they handling. Okay, next slide, please. Okay. Yes. So this is the World Health Organization guide to local production of the WHO recommended hand gel formulation. So we have adapted this procedure as during the early days of the pandemic last year. They have included. They have shown the actual what we call or ingredient for making hand rub, because there are other people who are making alcohol that sometimes that's not the proper way. So this guide, we have used this in our preparation. So next slide. Okay, sorry for that. Okay, so I am in the slide number 25. So yeah. Okay, so this is the reagents or for formulation 1 and formulation 2. They have formulation 1 and formulation 2, we have the ethyl alcohol, ethanol, and they have the isopropyl alcohol. So I just want to show it here. So can we turn into the next slide? Okay. We'll go to the next slide, ma'am. Okay, I'm not gonna read this anymore. I have just shown you the actual preparation according to WHO. So the next slide, please. Okay, this is just a flow diagram on how to prepare the hand mist, and it's just a simple one. So next slide, please. Okay, for the business opportunities, we are preparing this modality so that they could decide if they're going to venture in this kind of business. So this is just an assumption that they are going to have production days of from year 1 to year 5, 200 days in a year, 230 and 250. Actually, these are the like a feasibility study, economic indicators, and the daily production output of 500 units of hand soap, 200 units of liquid soap, and 200 units of hand mist with this price in peso. And all products were assumed to be sold for the entire 5-year operating period. Selling price shall increase by 5% per year. Cost of materials shall increase by 2% to consider inflation. Administrative and selling expenses shall be equivalent to 60% of annual gross profit, and all cost and profit calculations were based only from the laboratory scale of operation. So this was done by our economists. However, due to if we are going to a large scale, this will be a different scenario. So for the next slide will be the return on investment. Given these indicators, we have the discount rate of 10%, the net present value because of the assumption, as I have shown earlier, of 3.4 million, and the internal rate of return of 91%, and the return on investment of 121%, and payback years of 1.19. So this is just like a science going from laboratory to market, and we are in the pandemic situation. This is like an agile research, and we have done this for 6 months, and we have applied for the IPRs, and now we are waiting for the results for the dermal irritation test. So we have subjected the products for the dermal irritation test just in time before the before these products go to market, and all the regulations needed by the FDA we can provide. So that's our job at our institute, and we are promoting forest-based materials because we are mandated of the forest products. Okay, next slide. What is the next slide? Next slide, please, ma'am. Okay, hello. Next slide. Okay, so these are the updates. For now, we have different variants. So the SARS-CoV-2 variants, we have the okay, okay. So this is the research team that I have shown earlier. Okay.
Amita ma'am, your mic is off. Please turn on your mic.
Ma'am, please
Okay.
on your microphone.
inconvenience, but I'm almost finished in my presentation. Okay. Thank you so much, ma'am. Okay, the slide before this, can we,
Done.
yeah. Okay, so this is the research team that are involved in this endeavor, and we have respond to the needs for the proper hand hygiene. The group is composed of chemists, microbiologists, and engineers. So, okay, let me go back to the updates on the SARS-CoV-2 variants. We have the DG14C. So we have this is last year, January, February 2020. And we have the cluster 5 also. This emerged in August up to September 2020. And detected in December 2020 in UK is the B117. So this is the case of the Philippines because we have a biosurveillance laboratory. And then, okay, I have said that this is the research team. All millennials. So we have developed these products during the time of community quarantine. So, and limited budget, limited mobility, limited supplies. So, but we need to contribute and conduct agile researches like this. So, what's the next slide? I think the next slide is if you have inquiries. The next slide would be if you have inquiries, you can contact our director. Next slide, please. I think it's not moving.
I have shared next slide.
So, yes. Okay, it's delay in my side, maybe because of internet connection. So this is our address. If you have inquiries, if you have collaboration, we always talk about collaboration, the Prof, Dr. Amita and I. So this is our director and this is the address. You can contact us if you have if you want some inquiries and some collaboration with our institute. And then next slide. Okay, so I'm going to reiterate the observing of the COVID-19 protocols. And then, okay, to end this presentation, because this is a National Science Day and commemorating Raman, C.V. Raman. So I'm going to say that the essence of science is independent, thinking hard work, and not equipment. So you see, we are just using simple equipment. We are just using simple science here. When I got my Nobel Prize, I had spent hardly 200 rupees on my equipment. So this may be a simple one, but its impact on the pandemic is very high. So as simple as it seems, so I just want to share and inspire some of the students here that you can start to have simple thinking only, simple science. But because in simple things, you get the highest impact. So, and you get the highest application, especially in our nation, you can help and you can help develop also products and with our mandate of the forest product. So with that, thank you very much. Thank you for the invitation and happy National Science Day to all of you. Thank you.
Yeah. So, thank you, Jenny. Yeah, you have discussed about bamboo. In India, it is very common. Bamboos are available very commonly. But we have, you have done so much work on bamboo. And I think you have discovered so many products from bamboo and its application during this COVID pandemic. So thank you for your lecture, and I hope we will definitely collaborate with you. So
yes,
you.
we will have a collaboration.
Yeah, and in this short notice, you have accepted my request So after your after yeah I after discussion with you, then only I can yeah, I have planned this to celebrate this day after confirmation from you. So thank you.
Thank you. Thank you.
Yeah, thank you so much.
Thank you so much, ma'am, for your valuable insight. Thank you so much. Now, I would like to invite Dr. Ashok Kumar, presently working as Senior Research Scientist at Kansas University Medical Center, Kansas, USA. Uh, he has completed his PhD from University of Delhi in year 2013, M.Pharm from BITS Pilani in year 2002, Rajasthan, B. Pharma from College of Pharmaceutical Science, Manipal in year 2000, Karnataka. He has professional experience of more than 15 years. He has work experience as research associate at Kansas University Medical Center, Kansas City, USA. Postdoctoral associate at DA Medical Center at Brown University, Providence, RI 2016 to 2019. A postdoctoral associate at University of Florida at Department of Psychology and Functional Genomics 2015 to 2016, and many more works. And he is awarded doctoral scholarship, senior research fellowship by government of MCT of Delhi, India, 2008 to 2013. Get fellowship during post graduation. Sir, I would like to invite you for a few words on male sexual dysfunction and insight into screening.
Uh, Thank you, Shweta, for a very elaborate introduction. And since uh, Dr. Amita Verma invited me just yesterday, telling me the significance of this particular day, and I could I could recall that what I did my during my PhD days, that I was working on male sexual dysfunction. So I thought, like, since the audience of this particular seminar or webinar, you can say, are most of the B Farm students, so I would like to make it very simple so that most of the students can be benefited. So I'm, I want to share this. Uh, so you can see the first slide. So the topic of my presentation here today is male sexual dysfunction and insight into the screening. Here, the objective of this presentations will be to make you aware about the different conditions which are collectively known as sexual dysfunction. So sexual dysfunction is a very common problem in all across the world. It is a problem which is associated with females also, and also with the males. But in today's presentation, I will be dealing only with male sexual dysfunction. I'm not dealing with female sexual dysfunction. we would see that if we find out that some particular drug or some particular plant product which we can say from our traditional knowledge, or if some of the students who are very enthusiastic about synthesizing some compounds in their chemistry lab, and they found that, and they are having some confidence on those products that they will be very good for sexual potency. So the question is how to test that. How can they say that this product which I have synthesized, or the product which I have isolated from a plant leaf or from a plant bark, whether this compound is active and it can treat certain sexual problems in males. So that I will be discussing. And since I have screened two of the herbal products, which are Cuminium cyminium, which is commonly known in vernacular language in India as jeera, which is a common condiment which is used in our food, day-to-day food, and Tribulus terrestris, which is generally known as gokru, and it is a perennial herb which is commonly found in some dry areas of Western India. So these are my objectives of this presentation. Generally, when we say sexual dysfunction, which is a very broad term, we classify the sexual dysfunction into three different categories. Okay, the first category is lack of libido. That means the person does not have any desire to have sex. Okay. The second problem is organ dysfunction. That means the lack of erection during sexual intercourse. That means the person has the desire to have sex, but he is not able to do sex because of he is not having proper erection during the sexual intercourse. And the third problem, which is not very common problem, is oligospermia and aspermia. So the most common of these three problems is erectile dysfunction, or we can say organ dysfunction. All of you must have heard about a drug called Viagra. So that is the drug which is commonly given for this particular condition. I still remember the annual turnover of Pfizer, which manufactures this particular drug, was 18.2 billion some years back. And they are still selling it like hotcakes. Although the patent has been expired and a lot of other drugs have come into market, but this is still a very, very commercially a very, very viable area where many people can still venture out, since not many schools and not many books will educate you on these screenings. So I hope this presentation of mine will give you some idea about choosing your future branch when you are trying to see that what field you should go for. Okay, so dealing with the first one, lack of libido means the lack of desire to have sex. Okay, so why somebody XYZ person will have lack of sexual desire? There are a couple of reasons for that. One could be the low testosterone levels. There could be depression. There could be chronic illness such as diabetes. Somebody is suffering from diabetes, hypertension, obesity. He's not well, he's not healthy, or he's taking too much of stress due to work, or maybe any other reason, or must be having a lot of alcoholic problems, like he's been drinking too much alcohol. Then in these conditions, there are quite good chances that he may lose the desire to have sex with his partner. So suppose we develop a particular medicine, or we synthesize a particular compound, or we isolate a plant product and want to see that whether our product will increase the libido or increase the sexual desire or not. How do we check that? We check that before directly giving it to humans. We would check that in small animals, maybe rats, maybe rabbits, but the most common animal which are used for the screening is rats. So we will first develop a first we will develop an animal in which the sexual desire has been lost. Okay, so how we will kill the sexual desire in a rat? Okay, we will give him fluoxetine. Fluoxetine is a serotonin uptake inhibitor, reuptake inhibitor. So if we give fluoxetine to a rat at the dose of 10 mg per kg for 28 days, this animal is likely to get loss of libido. Okay, the animal will not be having any desire for having sex. And how do we know that whether rat is having a desire for sex or not? So I have developed a particular type of cage where I wanted to see that. So this is the cage which I developed. In this cage at the center point, I will put a male rat. Okay. Yes, that white portion, I will put a male rat. And in these two boxes, I will put a male rat, and here I will put a female rat. Okay. The female rat which I will put, I will inject estrogen and progesterone. So estrogen 48 hours before putting that animal into this box, and progesterone 1 mg per kg per rat 6 hours before putting that animal into that. Why we are giving estrogen and progesterone? When we are giving estrogen and progesterone to a female rat, the female rat comes into a heat. That means the female rat secretes certain kind of smell which attracts a male rat to have sex with her. This box is for male rat. This is for female rat. Here we are putting the rat which is having no desire. How we created no desire in that? By giving fluoxetine for 28 days. Okay. And now when we put female rat which has been given estrogen and progesterone, there are good chances that this rat will go close to this. So we will keep that rat in this particular chamber for 10 minutes. And out of these 10 minutes, we will count that how much time this rat will spend in this particular area. Okay. You may argue that this rat can go to this female rat only for social interaction, but not for sexual interaction. Okay, that is the reason why we kept a male rat here. Okay. If he want to socialize, he can go to a male rat. Okay, so we will try to find out of 10 minutes for which we will keep this rat in this center chamber, we will see how much time the rat spends close to female rat, which has been given estrogen and progesterone, and how much time the rat spends in proximity to a male rat. So this will be a social interaction, male to male, and this will be a sexual interaction from female to male. It is to be noted that the rat can never enter into this one because these chambers are separated by a wire. Where smell can go, the rat can see the other rat, but cannot go inside this chamber. Okay? So that's how we see that how rats can be created that they don't have any sexual desire. When we see that rat is not having any sexual desire, we give him the drug which we want to screen. Suppose you have some drug which you have purchased from some local market where they claim that, oh, this drug is very good, this can increase the sexual desire. You want to treat that rat with this particular drug and see that whether the time which it spends with the female rat is increasing or not. I hope I make it clear. So what we did is we calculated the time spent with the sexual incentive, that is female rat, and the time spent with the at the day 9, day 18, and day 28 at 3 different days. So when we were giving the fluoxetine, we could see that as the number of days increasing, the time spent with the female rat keeps on decreasing. Okay, this is the percentage of time which is spent by the rat which is in the center chamber with the female rat. Because as the fluoxetine dose keeps on increasing, the number of days on which the rat is on fluoxetine keeps on increasing, the percentage time spent with the female rat reduces from 40 to 20 to 9%. So that means if you are giving a drug to the male rat, it will not go to the female after 28 days, or the time which it spends with the female rat reduces considerably. But if you give cuminium cyminium, okay, which is jeera extract, we can see that the reduction is not there. Here 41 to 9%, it has considerably been reduced. But if you give jeera extract, it still spends a considerable time with the female rat. That means cuminium cyminium can block the fluoxetine effect. Okay, the other drug, tribulus was not very effective, and we can see a standard drug, bupropion, it also blocks the effect of fluoxetine. So this is the one way by which we can say that how to screen a particular drug, whether it is increasing the desire to have sex or not. So jeera in my case was found to be effective. Coming to the second, since I told you in the beginning of the slides that there are 3 different conditions which are commonly being associated with sexual dysfunction. The first one was loss of desire. The second condition is erectile dysfunction. Viagra is a common drug which is for this problem. So this is a condition in which a person is interested in having sexual activity, okay, but he is not able to perform. He is not able to satisfy his wife or sexual partner, okay, because the erection is not there. During the entire sexual intercourse, the person is not able to keep the penis in an erected form. So what are the reasons for that? So I broadly classified those reasons into 2 reasons why a person is not able to have the penile erection during the sexual intercourse. One of the reason is psychogenic and the other one is organic. When we say psychogenic, there are many reasons for that. Suppose somebody is not having good work culture at their place, or he's been bullied by his boss, or he's not happy with some of his colleagues, or some office tension he's bringing at home, then he may not be able to concentrate in the sexual intercourse. Similarly, somebody is not having a good relationship with his wife or sexual partner. They are not happy, so he may not be able to perform. So those psychological reasons are being classified into psychogenic category. Whereas there are certain reasons which are organic. So organic reasons means there is some medical problem which is associated. That's the reason why the person is not able to have the penile erection during the intercourse. When we see the most common condition in which the person is not able to have sexual penile erection during the sex is diabetes. Like if a person is having long-term diabetes, and that to uncontrolled one, he will generally have this erectile dysfunction. Or if a person is having long-term high cholesterol levels or he is smoking, there are chances that he may not be able to have a proper erection during the sexual intercourse. So, how erection takes place, you need to understand this first. So, this is the penile tissue. Okay. In the penile tissue, this is testis, and epididymis will take the sperm hair, and from that it will get ejaculated. So this is the penile tissue in a flaccid condition, and this is the penile tissue in a erect condition. You can see at the center of this, okay, if I cut this penile tissue transverse section and we see the transverse section from the top, we can see like this. Okay, so this is the vein which is on the top. Now you can see the blood arteries. These are the arteries. At the point where my arrow is right now moving, these are the arteries which bring the blood into the penile tissue. And these are called cavernosal arteries. These arteries will bring blood, and whenever the erection takes place, this area, which is called corpus cavernosa, is filled with blood. Okay, when it is filled with blood, these veins will get compressed. As a result, the blood will not be able to go to the body back. When it is not able to go to the body back, what happens? The blood gets engorged in this portion. As a result, the erection takes place. Why this? There are small balloon-like structures in which the blood gets filled. Okay, these balloon-like structures, they have smooth muscles on them. What happens in diabetes? These smooth muscles cannot get relaxed. Okay, when these smooth muscles cannot get relaxed, the blood cannot fill in them. So we need some drug which can relax the smooth muscles which are over these balloon-like structures. And Viagra does that. Okay. So Viagra relaxes these smooth muscles. As a result, the blood fills into them and the erection takes place. But how can we do this in animal? That is a tricky question. Okay. So what we do is, we take this penile tissue of a rat. We cut this tissue like this, take out this portion, which is like a tube-like structure. We take this portion from here to here from the rat, and this tissue, we will put it into an organ bath. I think this is an organ bath assembly which most of the students must have seen in their lab. Okay. So in this organ bath, usually the experiments which we see in our B.Pharm curriculum, we will put an intestine into this organ tube like this. And we will put some drugs into this tube, and we will attach this organ with a lever and see on the smoke drum that whether the contractions has been taken place or not. Here also, instead of an intestinal tissue, we will put a corpus cavernosa tissue, which I have shown in this slide, that from here to here, we cut a certain portion from the rat and we will put this into the organ bath. When we put this into the organ bath, we add some drug which can contract it. Suppose phenylephrine. When we add phenylephrine, there will be huge contraction. Okay, this is up kind of curve will be seen, and then we will give our drug which can relax it. Okay, for example, Viagra. We can add Viagra. Viagra will bring back this contraction back to the normal. Similarly, you synthesize some compound in your lab, in chemistry lab, and you want to see that whether it will relax or not. You can contract it with phenylephrine and then add your drug. If it relaxes, that means your drug is a smooth muscle relaxant. It is likely to improve the erectile dysfunction condition in rats. So I hope I made it clear. So what we will do, we will create the diabetes in rat by giving streptozotocin 60 mg per kg, and after 8 weeks we will see that these rats will have erectile dysfunction. How we can say that these rats are having erectile dysfunction? We take out their corpus cavernosa tissue, put them in organ bath, add phenylephrine, and then we will try to relax with our drug, whether our drug is relaxing or not. And we compare our relaxation with acetylcholine and compare the ED50. I hope pharmacology experiments, you know how to calculate ED50. Suppose this is our drug and this is acetylcholine. So which one is more potent? So this drug is more potent because the 50% relaxation comes with a very less dose. Here 50% relaxation comes with a very higher dose. On the x-axis there is dose, and here the pre-contracted tissue is getting relaxed. So this is the percentage relaxation which is coming. So this is how you screen for the second condition. So to recall, I told the condition of sexual dysfunction is in 3 different categories. First one is lack of desire. That means the person is not having a desire to have sex. For that I have shown you the chamber. Then the second condition is erectile dysfunction. The person is having the desire, but he is not able to keep the penile tissue erect during the sexual intercourse. And this is how in vitro you can check whether your drug can work or not. So here we can see in diabetic rats, tribulus terrestris, when it is giving, it causes the relaxation. There is a much more improvement of relaxation, which is 47% compared to the diabetic rats which were not treated with any other drug. There was a slight improvement here. Although the improvement is not as good as insulin, which is an anti-diabetic agent, but you can still find that the tribulus terrestris, which is Gocru, is having a slight beneficial effect. The third condition is oligospermia or aspermia. That means the person is having the desire. The person is having no problem with having sex with her partner, but he cannot conceive babies. Like, he cannot make her partner conceive babies. Okay, and that could be because of low sperm count. So WHO says that there should be 15 million sperms which should be present in each ml of our sperm. And if the sperms are less than that, maybe 5 to 10 million only, then the condition is called oligospermia. Aspermia is the condition where there is no sperm at all. And why there is oligospermia or aspermia? Because there are hormonal imbalances, testosterone levels are very low, person is having a long-term illness, or he's having certain exposure to pesticide, chemicals, or he has got some testicular injuries or some tumors in those reproductive organs, or obesity is also considered to be a major factor. And screening that sperm count is there or not, it is very simple. You must have got in your first year bachelor's of pharmacy, you must have seen Newber's chamber, and with the microscope only you can take out the sperm from the rat, from the epididymis, and if you dissect the epididymis, which is a connective tube between the testis and the urethra, there you take that epididymis and dissect it and dissolve in 1 ml of normal saline, and then you can with the help of Neuber's chamber, you can count the number of sperm cells in 1 ml. Okay, with the dilution factor and all, it is very easy experiment. Just like you calculate your count your RBCs and WBC, you can count your sperms also. So here we have seen the diabetes rats, when they were given cuminium siminium jeera extract, they were found to have increased sperm density. The sperm density is being reduced by diabetes also. So we have seen that the sperm density, when from a normal rat, diabetic rat has got very, very low sperm density. Okay, and this sperm density was to a certain extent recovered when we were giving 150 mg per kg of cuminium siminium. Okay, similarly we can calculate sperm motility also. With the Neuber's chamber it is quite difficult, but even if you calculate sperm density, that will be enough to have some basic idea about whether your plant extract or your chemical is working or not. So to finally conclude, among the two drugs which I screened, Tribulus terrestris and cuminium siminium, the sexual desire was increased by cuminium siminium and also the sperm count. And erectile dysfunction, the erection problem was to a certain extent was improved by Tribulus terrestris. So this was the part of my work which I did during my PhD, and I hope I was able to educate you in how to screen those compounds if you ever have a plan to do this kind of research. Thank you.
Thank you, Dr. Kumar, for sharing your valuable findings about male dysfunction and easy screening methods. You discuss in a simple way to understand erectile dysfunction and mechanism of various drugs in a very easy way. It is like your presentation was like in a very simple way, easily understandable. I hope his presentation will be very helpful to us in the findings of our further research regarding this field, STI. So thank you. And I think in US it is 2 o'clock approx, 2 a.m.
No, 12:30 only. I'm in the central zone and Hitesh is in the East Coast.
Yeah, I think Dr. Agarwal, yeah, I think, yeah. So thank you for giving your this valuable time. Thank you so much.
If you guys have any question,
yeah,
you can ask right now, or if you want to be in touch with me for any of your queries, you can have my email address from Dr. Verma, and she can help you in getting in touch with me.
Thank you so much, sir.
Okay.
Good night.
I request everyone,
good day.
please turn on your camera for a group photography, please.
Okay.
All of you, please switch on your cameras for group photo. Yeah. Imdad, please take one group photo. Imdad. So one more, Imdad, you can take.
Yeah.
Okay. Done? Okay. So now I would like to thank all on the behalf of Department of Pharmacy. You have done a wonderful job. So first of all, I would like to thank our speakers, Dr. Agarwal, Dr. Tamayo, and Dr. Ashok, for discussion about this STI and bamboo applications of bamboo products during this pandemic. So thank you all experts from US and Philippines. And I also want to thank our participants from India and Philippines. Thanks to all faculty members, dear students, for participating in this event. My sincere thanks to Mr. Imdad and Shweta. Both have done a wonderful job. We have discussed about this event. So speaker has also given their consent in this short notice. So thank you, speaker also. So Shweta and Imdad, they have done wonderful job. Imdad prepared all the poster and all the materials, and coordinated to all the students to join this event, and done a wonderful job and all. And thanks to Shweta also for anchoring. Thank you, Shweta. Thank you all to be a part of this event. I think we hope we will meet again in mid-March. I am planning one more event of American Chemical Society sponsored event, ACS event. This event is welcome, like about chemistry, create awareness about chemistry. And this is for school students and for bachelor's program. So I think we will meet again. Thank you so much. Thank you.
Thank you so
Thank you.
much, ma'am.
Thank you so much, ma'am, for giving me such a wonderful.
Thank you, Dr. Ashok. Thank you, Jenny. Dr. Agarwal.
So
Yeah.
congratulations.
Okay,
Thank
okay.
you.
Thank you, ma'am. Thank you.
Thank you. Leave. You have to leave this meeting.
Okay.
Or we can be like
I'm using the Google Meet for the first time. Thank you for
first time. Oh,
Yeah.
that's why. That's why. That's why. Yeah. So there's the option to leave the meet like red in middle to leave this meeting.
Yes, yeah.
Like,
Hello, Amita, ma'am.
Hello. Yeah.
Yeah, this is good,
Good morning,
afternoon. Yeah, good afternoon to you all.
Good
Good morning,
afternoon.
sir.
Yes, and this is a very interesting programs, and this is organized by the students as well as the Amita, ma'am. And yeah, I appreciate your efforts. And in future, yeah, I hope you will organize such more interesting programs to help us, yeah, in the scientific communities. So, so thank you, ma'am. So thank you.
Thank you.
Thank you, sir.
Thank you. Thank you so much. Thank you. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.